Okay. So we are looking now at another example of the application of um, the game theory principles. For example, we can have two sets of microorganisms. One are called the cooperative cells, the other one are called the cheater cells. Now, the cooperative cells are those cells which have the capacity of converting sucrose to glucose and fructose. And for example, the baker's yeast can do this. It can synthesize an enzyme called invertase, and it can break down sucrose into glucose and fructose, and this is of course happening outside the cell, outside the cytoplasm. Now, hold on, there are people waiting in the lobby. Okay, so now there are these cheater cells which are present, which do not have this capacity. Now, what do you expect when you have an ideal or a model system in which you have both cheater cells as well as the cooperative cells? Then what happens is that these cheater cells will usually grow much faster because they don't have to spend any metabolic energy for synthesizing this enzyme called invertase. And so they can grow faster because they grow at the cost of the cooperative cells, which are producing the invertase enzyme and then taking time to break down the glucose and fructose from the sucrose. And in the meantime, these cells being present in that environment, which are the cheater cells, are being present in the environment, start using up this glucose and fructose that is being produced by the cooperative cells. Now, this does not go on forever, of course, and if whenever the concentration of the cheater cells go high and the cooperative cells become low, and or when you are looking at low cell densities, when the environment is relatively not supporting growth because of other adverse conditions, then what you find is that these cooperative cells will actually thrive better because they are the ones that are able to convert whatever is present in the system from sucrose to glucose and fructose. So finally, it is always that these cells are going to win, but then you have these cheater cells which are going to have a problem. So this, when you model this system, you get these cycles which are produced in which you'll find cooperative cells first thriving, and then you'll have the microbial cells, the cheater cells, which are the cheater cells, not thriving in that cycle. They'll be in the other cycle. I mean, so you'll have the crest and troughs of these two groups opposed to each other. Anyway, so there are other ways to model it in which you essentially show that finally, no matter what the outcome, these cooperative cells are going to win, and the cheater cells are going to lose. And the next example that I'm going to show you here is the one that I talked about in the earlier class also, where we look at the growth of the tree and in a forest, and all of them start competing for the sunlight and the resources that are there on the ground, and so all of them will start growing at the same height, to the same height as far as possible, and there'll be some which will be having an extra height over the others, and these are the ones that are initially going to be gaining, and you'll find that over a period of time, because of these gain that they find, will get transmitted to the other trees, and so that will force all the other trees to grow to a certain height, and you'll find that is only advantages up to a certain height, which is called the H optimum. So beyond this height, you'll find that these trees will form a canopy on the forest, and you'll find only one or two trees which are extra in height because uh, uh, they are still competing for the resources, and they find that, well, there's no further advantage in going higher than that. So one or two trees will peep out of the canopy, but the rest of them will all be at the same level. And as you go further and further, the advantage that you get over having this extra height reduces further and further. So it's around this height that you'll find most of the trees that are present in a forest canopy. So these are principles by which you find that, well, evolution had other influences which converted the way biological systems started evolving. And besides the fact that at the molecular level you were seeing changes happening, which were happening with the genetic code and the survival of the species and so on and so forth, which were getting encoded within the organism, whether it be a tree or an animal, or a plant or an animal, and gradually over a period of time you find this huge biodiversity that is taking place on planet Earth. So So what are the factors that have shaped the evolution that we see here? One is the evolutionary factors, the other is the game theory phenomena, and the third could be dualistic features which are having a cost correlation related to the survival advantage. So there are these various other factors that are present, which are there. You have to analyze the system to really come up with these factors, and there will always be these two factors. One is the cost that is associated with it, and the other is the advantage that is associated with it. And based on these multiple factors, you find that systems evolve differently under different circumstances. So I'm going to end this previous lecture here and see if you have any questions to ask me on this right now. Anybody would like to ask questions on this so far before I go on to the new topic?
In this course, like we will be considering systems which are like systems as an ecosystem in which all these organisms and inanimate objects and things are interacting with each other, or are we just going to understand about genes and proteins and metabolites which are happening inside the cell? So
okay. So I mean, these first two, the first 4 lectures essentially that I've been giving. The first 2 lectures were introductory lectures. The next 2 lectures that I gave you are essentially showing you the basics of how the coding which was there within the cell evolved. So all of this has to be understood in the perspective of what we're going to be doing in this course of system biology. So the first few lectures were telling you the background and how the field developed, and the second part was to tell you and explain to you how the complexity was a factor that became so important for the evolution of the biological world. And within that framework, we have to understand what are the content. That means the genetic code, the RNA which was used for translation, the components of the cell that are participating in the cellular growth, as well as the enzymes and proteins which are further doing other things that are required for the cell. So those were the things that we were trying to understand. Then we also were, because we cannot isolate one organism and just look at the genetic code inside the cell, we also have to understand how this diversity and complexity took place at this macroscopic level also. And there what I'm saying is that besides what is there is happening at the level of the genetic code evolving and new things happening at that level, you also have other forces which are there outside in the environment, which also shape the way organisms are actually growing and competing with each other and then evolving. So there are 2 levels of things that are happening. But we will in this course focus primarily at the cellular level. We will not look at the ecological aspects of it, or we're not going to look at epidemiological aspects or the macroscopic changes that are happening or the evolutionary changes that are happening, but more to do with how do you address the changes that are happening within the cell, or how this regulation is taking place, and the way to express this in equations, and then trying to use that for describing what's happening. Clear?
yes, sir.
Okay. So what I'm going to do now is I'm going to now look at the next. I'm going to close this file. I think the other one is open. Close. All right. So I'm going to start the next lecture, and this lecture is the lecture on enzyme kinetics and metabolic pathways. And so I'm going to first start off with the motivation for doing this. So has anybody had a chance to look at the KEGG pathways?
Yes, sir. I opened the website, but wasn't able to figure out how to use it.
So can you see the pathway that I'm showing you now?
Yes,
yes, sir.
Yeah.
Yes,
so I'm going to I'm going to start off with this example. So if you just type in KEGG, let me see if I can go back to one more page and show you where I came from. So this is what you will see. Start off with. So this will be the database that you come up with. Okay. And so if I go to carbohydrate, metabolism, then I get this component which is here. And if I look under carbohydrate metabolism again, I get glycolysis. One of the examples of many, many examples that are there. So, so you can see that the way this is given is that you have, first of all, a class of reactions which are classified according to some known terminology we already know. So glycolysis is the breakdown of glucose to give you energy, and gluconeogenesis is the reuptake of smaller molecules to give you glucose again, or how you store, or how from storehouses of starch and other components you get glucose synthesized once again. So starch and sucrose metabolism, for example, it would be one of the metabolism from which you would be able to get glucose. And what they're showing you here is that you're getting alpha D glucose 1-phosphate from that source. Okay. Now, if this was to produce alpha D glucose, then there's an enzyme that is involved, and that enzyme is also given in this database. If you just click on it, it takes you to that enzyme. It is glucose 1-phosphatase, and the number is given to you. Now, if you want to go and look at other databases, for example, if you want to see what the reaction is, you can click on this, and it gives you the reaction, and it shows you that it takes alpha D glucose 1-phosphate, phosphohydrolase, and then phosphate, and this is the name of the enzyme. Sorry, it takes this, and then with the presence of water, so that's why phosphohydrolase, and it takes up and converts this into alpha D glucose and gives you an orthophosphate. And so the components are here with the code name numbers which are given to you. These are the codes for this group of compounds here. So C103 is D-glucose 1-phosphate, and then water is this one C401, and then alpha D-glucose is 267, and orthophosphate is 009, 409. So that's how this is showing you. And so it can go both ways. So if it goes in the forward direction, it takes up a water. If it goes in the reverse direction, it takes up a phosphate. So you can see that this is very well done, and it's an excellent website for starting off your work if you ever need to look at what's happening with the metabolic pathway. And let me go back here. So just likewise, suppose I need to check with a citrate cycle what is really happening with the citrate cycle. I can go here, and it will take me to another cycle, another page where the citrate cycle will be present. So this is the citrate cycle, which is going to start from this. It essentially starts with this pyruvate, phosphenol pyruvate, and then from the phosphenol pyruvate you get one arm that is going to go to pyruvate, and then from the pyruvate it enters into acetyl CoA, and then this cycle is the cycle where this pyruvate and has arm which goes to oxaloacetate, and then it goes on the citrate cycle, produces all of this, and comes back to malate, and then oxaloacetate. And you will notice that these are all reversible reactions, but there's a certain directionality of this whole reaction, and usually it goes down in one direction because it synthesizes ATPs as it goes down the cycle. Now, from this oxaloacetate, actually, you have a whole bunch of other amino acids being synthesized. Especially this one branch of this goes off into the aspartate acid branch, and from the aspartic acid branch you essentially get finally one other important compound, which is methionine. Okay, lysine, threonine, and methionine. That's what you get from there. All right. So this is the pathway, and you can see that these pathways, if I were to look at the way they have been constructed here, if I just look at this, for example. So from here to here, 6-phosphate, glucose 6-phosphate, there is a hexose kinase which will be the enzyme here. So there is an enzyme that is there associated, and there is a beginning point and the end point. And you see these are drawn as circles, often called nodes. And in this case, the nodes are the metabolites or the compounds. So you have this being the substrate, this being the product in this direction, downward direction, or the reverse, then this would be the substrate, that would be product. So substrate versus product is as what you can see here. So it shows you, and if I can go back to this one. And likewise, I'll go here to So there are other pathways, there are other places, modules also which it takes you. So it can also show you other databases besides the reaction ones. For example, previously it had a link to Brenda. Well, it's not showing me the link here, but you can also, there used to be a link from this which would take you to another site where you could see the structure of this enzyme also. But anyway, that's a separate thing that you can do in your leisure time. All right. Okay. Now, what I'm going to do is I'm going to.
I think similar to KEGG, there are other databases as well, right? Like I remember reading about a big database, BIWG, and a MetaPsych database as well.
Yeah, there are a few other databases. This is one of my favorite ones because it essentially has a huge amount of data, very well organized, and the other databases. This database is a very good starting point for making your search to understand what is known in the literature up to this point in time. It's regularly updated regarding even diseases also. So actually, I can show you. I do not know if there's a disease thing here. Human diseases, for example, right? And let us see if there's an Alzheimer's here somewhere. See, if you're going to cancer, something like huge, so maybe there are infectious, immune, neurodegenerative. Let's see, Alzheimer's. Okay. Now it shows you a huge bunch of, and not only that, it shows you what is there in the membrane, what is there in the neuron, what is there inside the cell in the mitochondria. So it has the classification by space also. So it's like tremendous actually. Also, there is not coming up here because sometimes they update the way it looks. So there should be another which is the main pathway. And that is not coming up here. So I don't know. I can just try this out and see if that is. I don't think it's going to go there. Let me just go back here. So we're looking at neurodegenerative multiple diseases, right? Let me just see if this one shows up as a no. Okay, this has become. I think what has happened is they have essentially expanded this further. So previously, it was much more easier to see the interaction of diseases. No? For example, this actually has grown since the last time I visited the site here. They've expanded this, so you can see for Parkinson's disease, you have one molecule here which is shown in red. The other one is in this one. In ALS, they're showing you TDP43, SOD1, and ATXN2. Prion disease, you have this. So you can see that in this whole space, the primary group of genes and proteins that are involved are shown right here. So you have kind of a summary shown here, but again, this is not showing you the interaction among them, and that's been expanded here. Sometimes it's very nice to have a smaller group of these proteins which are linked to each other, and you can see how they're connected and in which disease and how they are predominantly behaving, and then you can look at the details. So that I think somehow, maybe somewhere else, but they have relocated the way this is being done. Anyway, so the reason I am showing you that I showed you this the KEGG pathway is because you need to understand that there are essentially 4 layers of information that gets transmitted. Okay, broadly 4 layers of information. So what is the first layer? Do you have a whiteboard here? Let me see if I can go to the whiteboard. Breakout room. I don't see. So I might have to disconnect this and then go to whiteboard, right? I don't want to do that. Anyway, so let me just talk to you about this only. Oops. So doesn't matter. So what happens? I'll just try to explain this to you. So you have 4 layers. So first level is essentially the cell is isolated from the environment. You have a lipid bilayer which separates out the cell membrane from the cell's internal components, the cytoplasm, the mitochondria, and other organelles, from the environment which contains all that is happening in the outside world. And the cell has to respond to the outside world, whether it is isolated in a pool of water or whether it is part of a tissue. It has to respond because there are cues that are coming from other components, right? So that is what is called the signal. So there will be signaling molecules. In a tissue, it could be a hormone. In an environment, it could be a food or a toxin, right? Just something that bothers it or something that is that encourages it to move to have some nourishment. So the cells will have something that is there on the outside. So it has what is called a receptor. The receptor will receive a signal, and the signal that is received will get amplified within the cell as a secondary messenger. And the secondary messenger will then go forward and either, let's say, for example, send a signal into the nucleus from where you start synthesizing other molecules, right, or other proteins. Things like that can happen. Now you break this up into 3 or 4 different parts. So there is a signaling molecule. The signaling molecule will then go into the cell and it will activate, let's say, a gene. The gene will then get translated what is called a RNA. Then the RNA will then give you what is called a protein. And the protein will then convert something to something which is what the cell requires to act. So that's the action that is taking place. Now, the regulation that is taking place is also happening at every level. That means the signaling molecules are getting regulated, the receptors are getting regulated, the genes are getting regulated, the RNAs are getting regulated, and also the proteins are getting regulated. And the regulation is all interacting. That means at every level, from every level to every other level, there is a connection that is taking place. So when you see something like this, even if you break it up into 4 layers, the complexity of the 4 layers is quite huge. So to understand this, we have to start from the bottom. That means we go to the enzyme level and we start building up upwards and see what's happening as we go up from layer to layer. So at the bottommost layer is the enzyme, the proteins that are there. So let us say they have been synthesized. So what is really happening there? So what kind of regulation do they undergo? And what kind of categories can they be placed under? So the kinetics that we do is to see how they function. The regulation we do is also part of that because it will have the kinetics of that will tell us, because if there is an inhibitor, the inhibitor will change or alter the rate of the reaction. If there's an activator, that will alter the rate of reaction. And then they can be classified into different metabolic pathways, just like we saw in the KEGG pathway, that depending on where it is and how it is helping out in a sequence of reactions, it is classified according to that. So let us look at the primary function of an enzyme catalyzed reaction. And we need to understand this because we have to remember, and this has been done for some of you. You might have taken a course where you have done this. And the reason why we have to do this is because we need to understand how to represent this mathematically. So if you are building up a model, then we must know the background of it and how this model came about and how we finally use it. And later on, you don't need to know the derivation, but at least once when you're going through the course, you have to know how the derivation is done. So what we find is that in an uncatalyzed reaction, there is a huge energy barrier, whereas in a catalyzed reaction, the energy barrier is lowered. And obviously, because of that, in biological systems, you find that the reactions take place much more easily and at room temperature as compared to if you were to conduct this reaction out in completely isolated chemical environment. And why is that happening? Because by doing this, what is really happening? What is really changing? That what it means is that by lowering the energy barrier, so although these are of the same height, they're just shown that so that it magnifies the next part of the image. But essentially, this would be much lower than that. So if you have high energy of activation, then the number of molecules that would qualify to conduct a reaction would be much lower. So you can see that that would be the reason. Whereas if you have a low energy of activation, then a much larger population of molecules would actually qualify. Clear? All right. So let's look at the very first and basic reaction. How many of you have done this in your class? Hello? Somebody needs
Sir,
to.
we know about this equation, but not in class.
So have you done a detailed analysis of the enzyme of this, Michaelis-Menten reactions in at least those who are coming from DBB?
No, sir.
Not yet. Okay. All right. What about the others? Have you come across any of these reactions?
Yeah, I have been through this Michaelis-Menten reaction.
Okay. All right.
The kinetics.
Okay. Okay. All right. So let me explain this to you. Okay. Just look at this, and there is a whole method to this madness. So if you'll find as I go down 3 or 4 slides, this will get more complicated. Okay. What I'm showing you as a very simple reaction here. So, but you go step by step, and you can solve all the problems that come in the same category. So what I'm showing you here is a substrate and an enzyme combining to form an enzyme substrate complex. Then an enzyme substrate complex breaks down to give you the product and the enzyme back once again. The forward reaction is K1 and K2, K1 and K minus 1 here, and the forward reaction here is K2. Clear? So there are 2 derivations of the Michaelis-Menten kinetics. The first one is the classical derivation, where you assume that this first step, that is S plus E giving you the complex ES, is an equilibrium. So if that is an equilibrium, then what you find that this relationship holds. That means SE, that means S dot E, that means concentration of S multiplied by the concentration of E divided by the concentration of the complex is the ratio of these two, which is the dissociation constant. So that is the first thing that you must note. So that comes from the assumption. So whenever you are tackling a problem like this, you look at the assumption and then you write down the assumption in a form that is understandable by you in terms of these constants. The next thing that you have to understand is how do you write the rate equation. The rate equation is very simple. You just look at the product and you say that dp dt is the rate of change of the product, and that is the velocity of the reaction, which is nothing but the rate of formation here, which is K2 forward reaction, times the complex concentration ES. Clear? Then there's the balance equation that is there. What is the balance equation? The balance equation is on the enzyme. The enzyme which is there, which is the initial concentration of the enzyme E0, is assumed to be in sufficient quantity and constant. If that is true, then that concentration will be the free enzyme that is acting in this one after what is combined as a complex is present. So the free enzyme and the complex enzyme must equal to the total amount that was present initially. So these 3 equations are the primary equations. The thing to note is that this enzyme substrate complex that is there, it is something that you cannot measure or is very difficult to measure. So what you do is you essentially eliminate this from the set of equations. So that is the key point that you have to keep in mind. So when you look at this equation, then you try to eliminate this equation out. So this is nothing but from here, SE by K, and here I'm writing Km, that K is now the Michaelis-Menten constant here in this case. So turn out to be a Michaelis-Menten concentration later on. And that is nothing but equal to also from this equation E0 minus E, right? And then from here you get an equation for E, and what you do is you substitute back this equation into this first equation here, which is the rate of change of equation here, where you essentially write this out as dp dt is that ES, which is going to be nothing but this component here. So you put that down here, and then you just replace this E, because that is something that you can't measure, because that's a dynamic situation inside the cell or inside the reaction. And what you do is you replace that by a quantity that you know from a measurement, E0. So these two combined together gives you the Vmax. Now in this KEGG table, you will find that you can get for different organisms, the source that was used to get certain values are present there. And you can actually go back and look at the source, and you can get the K2 or the Kcat and the E0, which is the initial concentration, from the database itself. And that gives you the Vmax, which is dependent on the substrate. And so the V is dependent on the substrate concentration times Vmax and this one. Clear? Now you see this Km is nothing but this dissociation constant. So it gives you this value here. Vmax is this, and the summary of the Michaelis-Menten equation is given here. Any questions on this? Yes, no? At least tell me that.
No, sir.
Okay.
Also, no.
All right. So this is clear. Now we move one level up. Okay. Same set of equations, but you get what is called the Briggs-Haldane derivation. The Briggs-Haldane derivation is a more realistic derivation because it does not assume that this step is in equilibrium, but that this complex is at steady state. So the rate remains the same, which is based on this second reaction. V is dp dt, K2 times ES complex. The balance of the complex and the enzyme is equal to E0, which is the initial value, is already the same. And you look at the rate of change of ES dt, so that is going to equal to 0. But from here, it is going to be K1 times S times E, which is the first part. The second part is the negative part of this, which is K2 times ES complex, which is going backwards. Sorry, K1 minus K1 times E1, E2 complex on this one, and minus, plus K2. Sorry, the K2 is the forward reaction here, but minus K2 because it's removing this ES for when you look at this rate at which this is forming. So minus of that times ES. So that is the second equation here, third equation here. And you have the initial conditions which are present here. Now, based on this, you could eliminate this ES, and that ES turns out to be this value. And if you use this and go back to the first equation, you will get the rate equation, which is this one. And notice now that the Km has changed, and the Km value is now K negative plus K2 divided by K1. So that is the difference that you see. And in terms of what you see here, you will see that this one captures the initial dynamics that are there present in the enzymatic reaction. So this quasi steady state assumption which is taking place is actually holds here, and you can see that if you just not consider the initial dynamics that are present, most of the time you'll find that the enzyme complex ES is constant, E is constant, but the substrate is getting converted to product. And of course, based on the turnover numbers, you'll find X moles of substrate will give you X whatever moles of product, depending upon the stoichiometry. Clear? Now, there are several things that you could do. Now, notice that this can be integrated. This equation can be integrated. And if integrated this equation, then you'll get an equation like this, which is going to show you how, with respect to time, if this whole thing changes, the equation changes. So you could essentially have a plot that you could generate depending upon this S by S0 curve, which is versus the time, how this changes, you could actually work out. Now, this is an important exercise. You could perhaps do this in Excel. Normally, if you are having in-class lectures now, I would have actually asked you to do this on an Excel. So, but I'm leaving that out for now. The reason being is, this would just, I would not be able to watch you, and I wouldn't know if somebody's having a difficulty, and so I would not be able to help you with this. But I suggest that you please go back and try to at least work out this algebraic set of T versus S by S0 and see how this looks like. Okay. There are two other, there's one other point which I've not mentioned here. So if I go back, just give me just bear with me when I come back here. So this equation that you see here finally, which is V is equal to S by some constant plus S, right? Constant S by constant plus S, which is nothing but V max S by Km plus S. Okay, maybe this one shows it better. No, I've not done it like, yeah, right here. So look at this equation here and look at this last equation. So V is equal to V max S by Km plus S. This is a hyperbolic equation, and those who are doing mathematics also should note that this is a hyperbolic equation, where if you were to plot V versus S, then you'll have a hyperbola at minus Km and the other one at V equal to V max. Okay, and S equal to minus Km. And normally, all the simulation and all the results that we do are in the first quadrant of this hyperbola, where S is positive. So S cannot take a negative value. It is from 0 to some standard finite value that S can be permitted within a cell or in the environment of a cell. So whenever we are doing a simulation or looking at situations where we have to evaluate these constants using automatic methods of minimization, just be careful, because you have to restrict the domain of search to the first quadrant. Because if it goes into any other domain, if you don't do that, then what happens is it will go into another domain, which is the other part of the other three quadrants of the hyperbola, and give you incorrect values. And usually this simple equation then becomes a nightmare when you try to do any of the simulation work that is required, either for simulating a complex set of equations or just trying to do a minimization to evaluate the kinetic parameters from experimental data. Okay, a word about the plotting. So this is something also that students don't realize very easily, because it requires a little bit of experience in the lab as well as elsewhere when you're doing data analysis. So now in the lab, most of the time you'll find that the way the plot is done is 1 upon V by 1 upon S, and this is the Lineweaver-Burke plot. Okay, very standard plot which is done, and but you have to be careful. Keep in mind that that this is an amplification that takes place. So 1 upon S is an inversion, which, so as you get smaller and smaller values, which is usually where most of the time people are doing the experiments. So they will start coming up and appearing in this part of the curve, the data points on the outside, and the smaller values are not the ones, the reverse. I need to say the reverse. The smaller values are the ones which will come up with this part, and the larger values are coming at this part of the curve. So the larger value, the values that are normally in the experimental range are in this range here, somewhere there. And the ones which are done so that you essentially have more data points so that you can draw a line are on this range. But if you have experimental error taking place, then because of this amplification, you can actually tilt the curve of this, or use a regression where a certain value dominates and gives you an amplification of an error on this side here. And that is something you have to be very careful about. So look at the standard deviation that you get. If you have very high standard deviation, then make sure that you plot it in some other way, like in this one here, where you get a different kind of a plot where you don't have that dependency. This one also has a dependency. There's another plot which does not show that dependency. And this one also has some dependency. but there is another way to plot it where you don't have these dependencies. So the best way is to show that, let me just see this out of the way. So if you actually were to plot this one, so here you get a -1 upon k as the, so you have a v by s. So that is different. So essentially s by v by s. So this is not the same as this one if you notice. So this is s by v versus s. So this curve would actually have a line that goes in this direction. So it goes in this direction from here, and in that you will not see the separation taking place. To illustrate this, I actually have done a simulation here. For you to see. So you can see that when you do that s by v versus s graph, the data points are equally spread out. And if you had taken 1 upon v by 1 upon s, then you see that there's a separation of the data points here because of the amplification that takes place. And when you do the reverse v by s versus v, then you see also amplification taking place at this part of the curve. So this part of the curve will dominate the plot of this straight line, and this part of the curve will dominate the plot of the straight line. So if you have errors, then you have to be careful. Whereas this one, though normally it's not used, is actually equally spreading the data out, and you don't see any spread of the data. Clear? All right.
Yes.
Yes, sir.
Okay. So just keep that in mind that there are these 3 plots that are there, and normally the ones that are recommended or shown in the textbooks are not the best ways to do it if you are doing data analysis. If you have experimental data in the lab, there's not much you can do because sometimes you're bound by what is there in the data. Sorry, what is there, how you can conduct the experiment, the parameters that govern the experiment. But you could actually look at your data and be careful by having this knowledge a priori, that there is this chance of getting this problem. Okay. So the assignment that I usually give at this point is that whatever I showed you just now is plot s by v versus s in this range of km and this range of vm and this range of s. Okay, and you can do the similar plots for the other ones also. So please try this out when you are having free time now. So as soon as you get this uploaded, maybe, or this recording that you see a little bit later, please try to do this assignment. It's very important. Okay, so this is the first assignment. So you have to do this and then submit it to me online. Okay.
Thanks, sir.
Through email.
Yeah, just I don't know.
Okay.
Is there a better way to do it? Post it somewhere, and then I can pick it up from there.
So on Moodle,
So you could,
On Moodle,
yeah.
you can. Yeah.
Okay, I'll put it up on Moodle. Let me just frame it on Moodle so all of you can do it on Moodle and send it back. Huh? All right.
Yes.
It's a very easy
So one more thing.
assignment.
So should we plot it with some sort of like arbitrary data or just like using the equations itself?
Just plot it the way I've shown you here. No, just plot it the way the 3 plots that are there I've shown you. So 1 upon v by 1 upon s. So I mean I showed you the example is already shown to you actually. Let me see.
so would we be provided with some data points for plotting it? Or
yeah, I've given you already. See, the assignment is here. Plot. So this is the equation, Michaelis-Menten equation. Plot 1 upon v by 1 upon s, v by s by s, and sv by s for this range. So the range means I've given you a range of km values, given you a range of these values. So you have to take an s which is there between 0 to 10. So you can break it up into small values, like maybe 0.2 interval, something like that, and then plot that, right? Because automatically rest is dependent. So you can see the left-hand side will be dependent on the right-hand side of this algebraic equation, right? The v will be dependent upon s. So if s is defined by 0.2 interval, so you'll get straight away, maybe 50 points, data points here, 50 to 60. It won't be exactly 50, but so you'll get some data points here based on that, right? 0.2. So 5 data points plus 1. So 51 data points in total, I think. Maybe.
And so like KM and Vmax we can
Yeah,
specify.
okay, yeah. So you can change and see how that affects the result. So show at least 2 or 3 of these to see how this changes the slope. So you can see the change of slope that takes place. Right?
Fine.
You can take 2 values in each, take 2 values in this in the range that I've shown you. So anyway, I'll frame that problem and send it to you maybe on the Moodle. I've not used that, so maybe it's a good idea that I should also check it up. Okay. So what have we done for today? So I'm going to stop here. Unless you have more questions, I mean, I can go on. But I thought the next, okay. If you are willing, I'll do for another 2-3 minutes and just kind of go over in the next 2-3 slides, and then we can repeat that in the next class. Okay.
Yes, sir.
Okay. So I'm just going to do this because it'll just be one kind of set of things that I wanted to do. So the other thing that you have to keep in mind is substrate inhibition. And for substrate inhibition, there is this S that is there in the denominator, which essentially comes up if you have substrate as an inhibitor. So this happens very often when you have excess of glucose in a system. That gives you feedback inhibition to the system itself. So this is one kind of regulation. That if there's too much of substrate, then there is a slowdown of the rate of reaction because of the way the enzyme is constructed. It will not allow excess of the substrate to get utilized because you're spending too much of energy. So it kind of cuts down on the energy itself that is to be used, and it has sufficient means it doesn't want more. So and you can get this central point by just working this out and doing a dvds and setting that to 0 and taking the second derivative to show that it will be a maximum. It will have a negative sign, and then the value at which this happens is going to be square root of k1 k2. So that is something that you can just work out by hand and see what happens. Now, like I said, so there are 3 or 4 kinds of variations of this problem, and keep in mind the same technique has to be used. You first write out what is the velocity of the reaction. The velocity of the reaction is always with respect to P. So dp dt is equal to V. Then the other set of reactions, the other equation will be the equation that is the enzymatic balance. So what are the enzyme balance in this case? E, EI, ES is equal to E0. So that will be the enzyme balance. And then the other equation will be what is it that you're assuming here? So if you're writing ds dt is equal to 0, then what is the rate of change of ds dt? So des dt. What is the rate of change of that? Write that equation out and set that to 0. Now, then all you do is you manipulate it in such a way that you eliminate ES, the complex, and you eliminate E, which is the enzyme measurement, the free enzyme in the cell, and then you'll get the equation. So those are two main things that you have to keep in mind. So, so this one shows you that in this case, what we would get is a Km that is dependent upon the concentration of the inhibitor as well as constant of the. So this thing that you see here, the slope, the Km gets modified to a new Km value, and that Km value is dependent upon the concentration of the inhibitor as well as the dissociation constant of the inhibitor. So that comes up here, whereas the Vmax at the point where it intersects at the axis remains unchanged. And there is another one which is here, which is the non-competitive inhibition. And if you have multiple sites, but they're not competing with each other, then what happens is in addition to that variation that you see in Km, you also see a variation in this Vmax. And this Vmax is also going to be dependent upon the concentration of the inhibitor I. And that is so you see as the change in the slope taking place like this as before, but now the intercept is also changing because there's a dependence on the I concentration. I'm leaving it here. You can try it out and see if you can work this out. There is a whole series of these solved on the internet. Just pick up some of that and just see how this is supposed to be done. Try out at least this one. I think this one equation that you should try out. See if you can get the result that I've talked about here. The other one is a little more complex, but let's leave that out. But this is something that you could definitely try out, and it's not so difficult to do if you follow the principles that I told you to do. Okay. So what I'll do is maybe, I'll show you the solution of this maybe in the next class if I can work it out for you. Else you can just try it out and see. So I'll stop my lecture here today and ask you questions if you have any questions for me. And
So regarding the assignment, what is the deadline for it?
I think 1 week, no? Maybe if I can put it up by tomorrow, then you can just submit it by next Monday. Not Monday, next Tuesday.
Sure, sir.
Yeah, it's gonna be it's very easy to do. So
So we'll be doing it on Excel itself, right?
Yeah, Excel.
Okay, sir.
Okay. All right. Any other questions?
So can we use other like software, like not software, but other like methods to plot it, like Python or MATLAB?
Yeah, I think you can do anything you want.
Fine.
See, if you do, so the problem is, the thing is, if you do it in Excel, the plotting is very easy in Excel itself. If you do it, then I can kind of do it for everybody, grade it for everybody. But I mean, if you are comfortable with Python and other things, you can just send it to me. I'll have to again, what will happen is I have to upload the compiler and other things on my computer and then see what's happening.
Right, sir.
So prefer, at least for this one, I prefer if you give it on Excel. There will be other occasions where I will give you other options to choose from. Okay, because it's very easy.
Fine,
it's not difficult at all. This one is too easy. Okay. All right. So then bye for now. And see you again in the next class, which is tomorrow.
Thank you, sir.
Thank
Thank you, sir.
Bye.
Thank you, sir.
Thank you, sir.